Okay, so this is the third video recording of the neural cell development lesson. So neurons, when they are formed in the brain or in the spine, they need to project axon to connect to their target cells. These target cells can be nearby, so it can be projection to the dendrites to neighboring neuron, or they can be very far away. And in fact, some neurons project axon meters away, a meter away, or even more. So all this growing of the axon is driven by a structure which is called the growth cone. You can see here in this picture the body of the neuron and the axon that is growing, and at the end of the axon you have this growth cone structure. Here in this cartoon, here you have a representation of the growth cone. And in the growth cone we recognize a central region. This central region harbors microtubules, and these microtubules run all the way along the axon toward the cell body. We have an external region which is called the peripheral domain. In this peripheral domain, here is yellowish, we find this phylopodium which are rich in actin. So this phylopodium, like the fingers of a hand, are made of actin, so they have a core of actin. And you also have a transition region where you have not yet organized actin filaments. So the region of the growth cone that actually does most of the work are the lamellopodia. Lamellopodia are the region in between the phylopodium. The lamellopodia can, if you want, could correspond to the palm of your hand. This palm need to bind to the surface, in this case the surface will be other cells, and push the rest of the axon toward it. On the other hand, the phyllopodia, at times also called microspikes, the job of the finger is not much to grab like it would be in a hand, but is to sense. So these phyllopodia have a lot of sensory receptor, and they sense the environment, so that they can turn the axon toward one direction or another. So they can turn the axon toward stimuli that are positive, attractive stimuli, or away from stimuli that are negative, so deleterious or negative stimuli. So the job of the phyllopodia is to sense the environment and direct the growth. Now, toward the direction of growth, new microtubules are produced or are synthesized or organized in the central domain. In this way, the axon actually is grow toward the direction that the phyllopodia are sensing. So the entire axon body is extended in the direction of growth of the microtubules, and the direction of growth of the microtubules is determined by the phyllopodia sensing around. If we look at the tip of a phyllopodia, like in this picture here, we find at the end of a microtubule proteins that are often referred to as positive tips proteins. This is because they bind at the end of the microtubules. And these proteins are some example of this protein are CLASP or APC. Here in this cartoon you have an example of APC. These proteins are important because what they do, one of the things that they do is that they control the synthesis of tubulin from mRNA. Tubulin obviously is the protein that make up the microtubules. mRNA is produced centrally in the soma of the neurons that can be very far away, and then the mRNA migrate all the way up to the tip of the philopodia, where they can be translated from ribosomes that reside there. And this translation into protein is coordinated by this APC or class protein. However, the final extension of the philopodia of the finger is made by filament of actin. And actin can be synthesized or degraded depending on the environment. So raw GTPases like RAW-A, RAC1, and CDC42 mediate the response of the fingers of the philopodia to the external stimuli. The most important stimuli that these raw GTPases respond to are epinephrine, nephrine, slit, and semaphorin. So depending on which stimuli are around, new actin filament is produced, or the filament that was there is degraded. So if it is produced, the philopodia extend, so the axon go in that direction. If the actin is removed, the philopodia retreat, and therefore the axon goes away, goes in another direction. And if you want, in this cartoon here, we have a representation of what's going on inside the philopodia when they encounter the different stimuli. So the different stimuli are represented here. They are external to the philopodia. They are ephrines, they are netrins, slit, and semaphorin. Those bind a receptor on the axon. One important thing to notice is that not all axons, not all philopodia have the same receptor. So time to time, a philopodia does not have a receptor for a particular external stimuli, so they don't respond to that stimuli. So even if it is a negative stimuli, if they don't have the receptor, they are not going to respond to the negative stimuli. We are going to see an example of this later on. Now, when these receptors are activated, different pathways inside the cells are active or not. So we have activation pathway or repression pathway involving RAW-A, RAC1, and CDC42. For example, RAC1 and CDC42 are mostly involved with the synthesis or polymerization of new actin filament, so growing of the philopodia in the direction of the stimuli, while other stimuli like RAW-A is mostly involved with disassembly of the actin filament. So if you disassemble the actin filament, we are saying the philopodia retreat. If the philopodia retreat, the axon move in another direction. So how the axon guidance actually work? Well, the axon guidance has three important phases, if you want. The first one is pathway selection. During pathway selection, the axon travel toward positive stimuli and away from negative stimuli, and more or less the axon move toward the tissue that it should reach. So here in this cartoon, you see how the axon, thanks to the growth cone, is moving toward a region where you have a positive stimuli, and then move away from a region with a negative stimuli, and move away from a second region of negative stimuli here, and then reach or try to reach the target tissue. Once the axon reaches the target tissue, we have target selection. So when you reach the target tissue, the axon start to bind the cells on that target tissue. And the initial binding can be very generic. It doesn't need actually to be related with any axonal activity. In fact, these first two elements, the pathway selection and the target selection, do not require any biochemical or electrical activity in the neuron. The last part is called address selection. In address selection, the initial genetic connection to the tissue becomes very specific. And the axon bind, instead of binding generically many cells, start to bind only few target cells. And this third part depends on axonal activity. So if the axon is not actually used, so it's not biochemically used, it will not form strong address. It will not form strong binding with the cell and will eventually die by apoptosis. And we will see this in a minute. One good example of axonal growth relates to the axons that innervate limbs. So both motor neurons and sensory neurons project their axon toward a specific, for example, a specific muscle. They bundle together and they form what is called a fasciculate. So they fasciculate together, form like a bundle. They project together to the same region. And you can see in this cartoon here the projection toward a limb bud. So this is a developing limb, and you can see that the neurons from the spine organize together in fasciculating bundles, projecting probably toward the same muscles in the limb. Now, how these neurons project is already predetermined even before the axon starts to extend. And it is predetermined by OX genes expression and limb gene expression. So OX genes and limb genes control the direction of the initial direction of travel, of these axons. For example, an example of limb genes that are involved in this are islet1 and 2. If a neuron expresses only islet1 and 2, it will project in the limb bud ventrally. So you can see this one here. The limb bud, in fact, is divided in a ventral and dorsal region. The neurons that project ventrally, because they express the receptor for semaphorin 3F. And semaphorin 3F, it is only expressed in the dorsal part of the limb bud. So the neuron that express islet1 and 2 cannot actually go there because they are sensitive to semaphorin 3F inhibition. Conversely, neurons that express islet1 and 2 and limb1, which is another of this limb protein, can project only toward the dorsal part of the limb bud. You can see this here. And this is because limb1 induced expression of a receptor for ephrin A5. And ephrin A5, it is only expressed in the ventral part of the limb bud. So these neurons expressing the receptor for ephrin A5 cannot go there. They're sensitive to actually ephrin A5. Finally, we have a third group of neurons that is interesting to mention at this point, which are the neurons that express islet1 and 2, but also LHX3. LHX3 induce expression of FGF receptor in these neurons. And FGF actually attract neurons if they express the receptor. So those neurons do not project toward the limb, but they actually project dorsally toward the dermomyotome, which is at this moment one of the source of FGF signaling. So generally speaking, we have endocrine factor or elements that attract neurons long distance and short distance, or repulse neurons long distance and short distance. Some of the long distance clues that attract neurons are represented by nephrins, and some that repulse them are semaphorins. On the other hand, cadherin attract neurons on a shorter range, while epinephrine inhibit neurons on a shorter range. So uh, an axon that experience nephrins will move toward the source of nephrins and move away from the source of semaphorin. And similarly, it does with cadherin and ephrins. Now, here I come back to this cartoon that I used to show you how the neural crest cells only migrate to the dorsal region of the sclerotum. Similarly, the motor neurons only migrate or only grow through the dorsal region, through the anterior region of the sclerotum, not the posterior region of the sclerotum. Because the posterior region of the sclerotum express epinephrine and semaphorins, we already saw that, and these inhibit are elements that inhibit neuronal or axonal growth. But how the axon finally target the correct group of cells or the correct tissue in the correct cells in the correct tissue in the body? Well, this is mostly due to expression of several different kinds of chemotactic proteins that are released from the target cells. These include endothelins and neurotrophins. There are two modes of sensitivity to these proteins. One is defined as analog and one is a digital mode. In an analog mode, the sensitivity to the particular protein depends on the concentration of that protein. So if you encounter that protein at a low concentration, the attraction is very low. At a higher concentration, the attraction is very high. Or it can be the other way around, it can be repulsive. Other molecules function in a different way, in a digital way. Either they attract or repel or not. So if they are there, almost independently of their concentration, there is an immediate attraction or repulsion of the axon. And if they are not there, of course, they don't work. An example of analog mode of attraction is represented by the neurons that from the retina of the eyes of the vertebrate reach their target region in the back of the brain. So here we have a representation, a cartoon of a bird, and we have the retina from the eyes that produce neurons whose axon need to go into the posterior region of the brain, which is called the tectum. This tectum is a region of the brain that important for processing visual information. Now we have, because the retina is three-dimensional, so the retina is organized around a semicircle. We have neurons that are dorsal, neurons that are ventral. We have neurons that are more toward the nose, so nasal, and neurons that are more outside, so temporal. All these neurons project in different regions of the tectum. How do they do that? Well, for example, the retinal dorsal neurons express high level of TBX5, which is induced by nearby BMP4 signaling. And this very high level of TBX5 induce the axon to actually bind the tectum in a ventral region. On the other hand, the ventral neurons have high expression of PAX2. And this PAX2, which is induced by retinoic acid, allow connection to the dorsal tectum, so dorsal region of the tectum. This, if you want, is an example of a digital mode. High level of TBX5 or high level of PAX2. giving you different target site in the tectum. However, neurons from the retina also are an example of analog situation. In fact, the posterior cells of the tectum express effrins, and these effrins produce a posterior to anterior gradient along the cells of the tectum. Now, the axons and the neurons in the different region of the retina express the effrin receptor at different level. For example, the nasal neurons and therefore axon express very low effrin receptor. Because they express very low effrin receptor, they are not very sensitive to effrin itself, so they are not much inhibited by effrin, and only very high level of effrin inhibit their growth. So they bind into the tectum, they bind deep into the tectum in the most posterior part of the tectum where there is very high level of effrins. On the other hand, neurons from the temporal side of the retina express a very high level of effrin receptor, and therefore those axons with very high level of effrin receptor cannot go very deep inside the tectum because they are very sensitive to even low level of effrins. So they reach the tectum and only bind there in an anterior position. So, depending on the level of expression of TBX5, PAX2, and effrin receptor, the different neurons in the retina by different dorsoventral and anterior-posterior region of the tectum. Finally, when the axon reach their target tissue, we have the formation of synapses. So this is what we defined before as address selection. So synapses are a specialized junction between the axon and their target. Here we have an example in this cartoon. We have an example of synapses between an axon and muscles. So initially, the presynaptic axon release neurotransmitter that change the polarization of the membrane of the cells of the target tissue, in this case, the muscles. And initially, there is formation of unspecialized contact. So what we have here is that, for what we can see in this cartoon here, is that the axon is releasing agrin, and agrin cause or induce the cells of the muscle to cluster acetylcholine receptors. Now, after that, there is an accumulation of neurotransmitter in between the axon terminals and the cells of the muscles that induce the formation of a laminin-rich matrix. This laminin-rich matrix seems to be a specific stop signal dedicated to muscles. It's like the muscle is telling the axon to stop there, so through this laminin signal. What's happening at this point is that when one axon starts to form a connection, we have that other axon converge to the same place and start also to form connection. So we have what is called a multiple axon. So we have what is called a polyneural innervation, because multiple axons converge to the same place to form to the same synaptic site. However, what is very interesting is that after birth, most of these extra neurons die out, and at the end of the day, only one synapses, only one neuron survive. So only one axon having an innervation and a synapsis survive, and the rest are eliminated. And this process of elimination depends on activity. So depending on which axon is the most active, that one will survive, and the other ones are removed. And this process seems to be correlated or associated with production of nitric oxide. So the most active axon will produce nitric oxide that is going to induce apoptosis in the ones that are less active. And in fact, programmed self-death or apoptosis is an important element in how vertebrates build their brains. For instance, in humans, we produce 3 times more neurons that actually we have at birth. So 2/3 of the neurons are going to die by apoptosis by the time we are born. Apoptosis is programmed self-death, which is very different from necrosis. Necrosis occurs when there is a damage and the cells cannot be saved, so they die out. Apoptosis, on the other hand, is programmed in the cells. It is used very often in developmental biology. The classical example that everybody does or explain is the disappearance of the membranes in between fingers. This membranes is there, of course, in some animals that are aquatic, and for example amphibian, but is lost in animals that are not aquatic, like us. And time to times, because of some error during development, children are born with some of this membrane not disappearing. So we actually have individuals with membranes in between fingers. How apoptosis is controlled? Well, apoptosis have been well studied in nematode, and in nematode, which is this worm here in this picture, is very small worm in this picture. We know that all the cells in the nematode are already programmed to die, and the only reason why most of these cells do not die, most of the nematode cells do not die, is because they express SED9. SED9, in fact, blocks the expression of SED4. And SED4 is a protease activation factor, and when it's active, induces the expression of proteases like SED3. And as the name suggests, proteases are enzymes that chop down other proteins. So they chop down all the protein of the cells, inducing apoptosis. Humans' program, apoptotic program, is very similar. Actually, it's almost identical. It starts with BCL2. BCL2 is actually an inhibitor of APAF1, and APAF1 is a protease activator factor. So BCL2 is the homologous of SED9, and the two proteins are so similar that we can actually take the protein from humans and put it in nematode, and they will do exactly the same job. APAF1 is very similar, is an homologous of SED4, is a protease activator factor. When it is active, it induces caspase 9 and 3, which are proteases that chop down all the protein in the target cell. So a lot of the neurons that are not the neurons that do not survive the last part of synapses formation are killed by apoptosis, and in those neurons BCL2 is stopped, is blocked, so APAF1 can be activated, and therefore caspase 9 and 3 chop down all their proteins, and they go to the process of apoptosis. Okay, so the objectives for this lesson is to be able to explain how the brain develops from neurogenesis, discuss the contribution of neural crest in the development of the nervous system, and explain how axons grow and find their own targets.